In the last lecture, we considered the driving forces for molecular motion. And what I want to do in this lecture is return to a higher level of organization and a consideration of tissues. And in particular, we're going to talk about epithelial tissues and how the epithelial cells create membranes and then act as gatekeepers for the molecules that are going to enter into your body. And epithelial cells, much like connective tissue, comes in a variety of forms. And you see that there are functional differences in those different forms, which appear in different regions of the body. So just to give a little bit of an overview here, we have these various types of epithelial membranes. So we have protective epithelial cells. This is many layers of cells that provide a structural and a chemical barrier. So for example, your skin has this function. And in that case, the case of the skin, the goal is to keep all molecules out, right? The only molecules we want entering the body are the ones that we, inspire through our lungs, or that we eat and we deposit into the digestive tract. And there are different types, which we'll get to in a second, of epithelial cells that act as gatekeepers in those two organ systems. The skin just wants to keep everything out, okay? We want to instead allow for those other types of epithelial cells to serve as the mediators of molecular motion. And so that brings us to exchange cells. Exchange cells we have be in the lungs and in the endothelial lining of the capillaries. And the goal there is to overcome the challenges posed by Fick's law. So it's helpful to have these cells be very thin, and they might control the flux of molecules to some extent. But the idea is to be very porous. We have transport epithelial cells. So these are the ones that line your GI tract. And this is a single cell layer that controls the movement of molecules between apical and basal surfaces. In our example that we're going to feature in this lecture, we're going to talk about the transport of glucose. So we'll get to that in a second. We also have secretory cells, which secrete stuff. So they release chemicals like mucus, they release hormones, they play essential roles in the endocrine system, which we'll get to later on. They just secrete stuff. So they're not in the business of taking on molecules across the membrane, but they're just releasing molecules. And then finally, we have ciliated cells. We'll talk about them a little bit. You have cilia in the fallopian tubes and the sex organs of women. We have cilia that line the trachea and the other air passageways within the lungs to keep particles from accumulating in the more delicate exchange surfaces within the lungs. But our focus on this lecture is on those transport cells that line the GI tract. And the question for this lecture is, how do digestive, that is the epithelia within the digestive system, how do digestive epithelia transport glucose? How, when you eat a meal that has glucose, which is a precious nutrient, even though we're showered with it in our American diets, how is it that we can take on the glucose and maybe not absorb molecules that we don't want to absorb? And we're just using this as an example of how you have an epithelial cell that's controlling, it's really harnessing the driving forces of molecules in order to get this good stuff into the interior of the body. And so, like we often do, we'll consider a sort of a simple model where we have, you can imagine this interstitial fluid, that is a fluid outside of a cell, being within the GI tract. So this is all a set of molecules that have emerged from the meal that you ate, and then it was broken down by enzymes. So in this case, we have a bunch of chloride and sodium ions that we're focused on here. There's, of course, lots of other stuff you get from meals. And then on the right-hand side, we have what's inside of a cell. And so we already considered a version of this in the last lecture. On inside cells, you tend to have proteins. Proteins have a slight negative charge. We also tend to have a high concentration of potassium as well. Now, what we have here is a separation of different kinds of molecules. So you have the chloride and the sodium ions on the left-hand side. You have the potassium ions on the right-hand side. And of course, molecular diffusion will favor the potassium moving to the left because it's going to want to go down its chemical concentration gradient, and for sodium to move to the right. That is the natural tendency. If we were to just open up the floodgates and to allow all molecules to go across this membrane, then chemical diffusion will tend to have the concentrations be equal on both sides of that membrane. Now, there's a tendency that your cells have to counter that. It is beneficial, as we will see, to have a lot of potassium on the interior and to have a bunch of sodium on the exterior of the cell. And this is so important that our cells invest a significant amount of energy to keep these concentration gradients maintained. And the source of energy that we use within cells is to take adenosine triphosphate, to hydrolyze it to adenosine diphosphate. That releases energy. And what we do in the case of this transmembrane protein, which is an example of primary active transport, is we do that hydrolyzing of ATP to ADP. And in the process, we transport sodium from the interior of the cell to the exterior. And then we do the opposite. We transport potassium from the exterior and stuff it into the interior of the cell. Because we're moving against the chemical concentration gradients, that process requires energy. And that energy is harnessed by this transmembrane protein known as sodium potassium ATPase. This is an example of what's called, this transmembrane protein is an example of what's called a carrier protein. So these are transmembrane proteins that change conformation to move molecules. Okay, well, you can assume all cells have sodium, potassium, ATPase in this course, and they will act to create this chemical concentration gradient. We'll see lots of cases where this plays a critical role in how a cell functions. Another transmembrane protein that is common to cells is one that undoes half of the work being done by sodium potassium ATPase. And that is the establishment of the potassium gradient across the membrane. What is this transmembrane protein? It's known as a potassium pore or a leak channel. And because chemical diffusion favors potassium leaving the cell, this doesn't require any energy. You can think of this as essentially just a hole in the cell membrane that just allows only potassium to cross. It's known as a channel protein. So channel proteins do not require hydrolyzing ATP. They just use facilitated diffusion to allow for this movement across the membrane. And it happens to be a water-filled opening. But the important property here is that it's not requiring any energy to do this. Now, you might wonder, why would you want to invest the energy to move potassium one way only to have it undo itself with these guys? And to answer that question, that will come when we talk about the membrane of an axon. So you'll just have to store that mystery away for the moment. Because our current focus is on epithelial cells. Here we have in your small intestine 3, or at least part of 2, and 1 single cell of one of these epithelial cells. And this is highly schematized, but we imagine that the gut lumen, that is the interior of the gut, is up top. And then this space between the epithelial cells and surrounding capillaries and other tissues is our interstitial fluid. So You could say the interstitial fluid could correspond to the space in the gut lumen. In this case, we're distinguishing the gut lumen is where the nutrients are going to be, and the interstitial space is where those nutrients are going to get transported to then get picked up by the capillaries. Now, in order to take a bunch of cells and to stitch them together so that they provide a membrane that has some structural integrity to it, but also doesn't allow molecules to sort of slip between the cracks here, then we have two types of structural proteins. First, there are desmosomes. Those are the ones that provide a structural reinforcement between the cells, so the cells don't just break apart. You can also imagine that there's a large extracellular matrix with collagen fibers as well. And then we also have a chemical barrier that's provided by tight junctions. So you want to remember the difference between desmosomes and tight junctions, but the overriding point here is that we can, through those proteins, stitch together cells to provide a membrane. All right, so here's the goal. We want to take the glucose and somehow get it from the top, from the gut lumen, into the interstitial fluid. Here's the challenge. Chemical diffusion really does not favor this process because when you look inside of any cell, glucose is an important precursor to producing that ATP that we just talked about. So when we talk about cellular metabolism, which I'm sure you have some familiarity with, we're going to convert the glucose into ATP. But the bottom line is that cells have glucose at the ready to produce ATP as needed. And so there are circumstances in your GI tract where the glucose inside the gut lumen can be a lower concentration than inside the cell. That's the challenge. How do you get the glucose to move against its chemical concentration gradient so that it traverses our epithelial cell to make it to the interstitial fluid? This is where sodium potassium ATPase comes in. And in this context, just like I've outlined, it functions to do that. It just creates a potassium gradient and a sodium gradient across all membranes of the cell. That is the apical membranes and the basal membranes. And so what that means is that on the apical surface, that is the surface up here, you can imagine that there's a higher concentration of sodium ions on the outside of the cell compared to the inside of the cell. Because chemical diffusion favors sodium rushing into the interior of the cell, you can think of this almost like a dam that has a certain amount of potential energy. And then if you allow the dam to open, you can capture some of that energy as it moves down its gravitational gradient. Here, this separation of sodium ions from the exterior into the interior is similarly a source of energy. And so if you allow sodium to move down that chemical concentration gradient, then you can potentially capture that energy to do something else with it. And before we get to that, just note that the whole reason that we have this opportunity is that sodium potassium ATPase is investing ATP to establish that chemical difference across the membrane. Okay, how do we capture that energy? Well, on the apical surface, we have a transmembrane protein, again, that grabs onto a sodium ion, that is the source of energy, and it also binds to a glucose molecule. And once it has bound, excuse me, once it has bound to both of those molecules, then it will capitalize on the sodium energy in order to transport the glucose against its chemical concentration difference across the membrane. And once the glucose is in the interior of the cell, then it's free to move down its chemical concentration difference on the basal side of the cell, and because it doesn't require any ATP, because that is a favorable direction in terms of the concentration difference across that membrane. So what we've done is we've invested energy through sodium potassium ATPase. we've capitalized on that energy through this transporter up here. And then it's just a small matter not requiring any energy to allow that glucose to then leak out into the interstitial fluid. So this features carrier proteins. So we already described sodium potassium ATPase as a carrier protein. This particular carrier protein uses primary active transport. That means it has the capacity to hydrolyze ATP. In contrast, our red transmembrane protein here, known as a sodium glucose symporter, uses the concentration gradient established by sodium ATPase as a source of energy to transport the glucose against its chemical concentration gradient. And that's what's known as secondary active transport. So it's not directly hydrolyzing ATP, but it's just using the product of ATP, that is the sodium gradient, in order to transport glucose against its concentration gradient. And then finally, we have this glut transporter, which just uses facilitated diffusion to allow the glucose to transport. So you see among carrier proteins, there are three ways of generating and capitalizing on energy in order to transport molecules. So the question that we laid out at the beginning was, how can a membrane control molecular flux? We have these carrier proteins, which we can categorize by how they use energy or don't use any energy at all. And then we can contrast that to the channel proteins that I mentioned. So we had that potassium leak channel. Channel proteins are also super, super important. They all use facilitated diffusion. They have the fact that it's a water-filled opening is another sort of property, but really we're focused here on energy. So they always use facilitated diffusion. You have pores like the potassium leak channel, which are always open. And then there are also things known as gated channels, which we'll itemize later, which are triggered to open. So they have an ability to vary their permeability to a type of molecule, whereas pores are just always on. They're always open and always creating high permeability to a particular type of molecule like potassium.